Dear students, welcome to the last part of the recombinant DNA technology. That's application of recombinant DNA technology, wherein people are using this particular method in developing varieties of organisms, proteins, cells, etc. So, first, in production of recombinant DNA, recombinant proteins, by using this particular method, proteins that results from the expression of recombinant DNA within a living organism or cells is called as recombinant proteins. So, in production of recombinant proteins, so it is being done very mm, in case of bacteria. They are producing it in case of yeast cells and insect and mammalian cell lines. So in order to produce different type of proteins, we need to have an expression system. There are four different expression systems people are using. Bacterial, wherein they are using plasmids and phages as vectors. In yeast, they are using expression vectors like plasmids, yeast artificial chromosomes. And insect cell lines, where baculovirus and plasmids are being used, and mammalian cells, where varieties of viral vector systems are used. The most stable cell lines which are being used in the mammalian is CHO cell lines and HEC 293. So these are the expression systems. There is cell-free systems is there, and even people are using the organism also in the production of a particular protein. So in order to express a particular protein in an organism, we need to have a specific type of vector systems called as expression vectors. These expression vectors are going to have a key characteristics which are going to be used either in bacteria or in case of eukaryotic cells. The characteristics are that it should have an in vivo promoter which can induce a sense of particular mRNA by the gene construct. There should be a selection marker. There should be a sequencing primer which can be used for sequencing. The expression vectors are going to be used to produce specific protein in large quantity. It permits the study and structural functions of proteins, can be used when proteins are going to be rare components of a particular cell, or whenever it is difficult to isolate a particular protein, people are going to use this method to produce it in large quantity. So there are three different types of expression vectors. Transcriptional vectors, so wherein these particular vectors are going to be mainly used to get more mRNA. There are translational vectors, and which are going to be used in order to produce these particular proteins within the cell. There are translation vector Placidia, which are going to be placing these particular proteins in membranes. Then expression system wise, bacterial system, it has some advantages: fast growth, cheap medium, equipment for growing, and good knowledge is there about the host. So some of the common problems with bacterial system, we have a problem with the change of promoter, change of plasmid, change of cell type. All these are going to cause low level expression of the protein, and there is severe protein degradation is also going to be seen in bacterial system, because when the protein is going to be non-bacterial host, non-bacterial origin, there are eukaryotic origin, so then there is a chance of degradation, so which is being solved by using proteasome inhibitors and other protease inhibitors. They are using it, and even they are trying to induce low temperatures based components. And there is again problem like missing in the post translational modification, very important restrictions in the case of expression of eukaryotic proteins in prokaryotic system. Glycosylation again is a difficult process in case of prokaryotic system. To solve this, whenever a highly glycosylated protein need to be synthesized, people are going for yeast or mammalian system. Another very important problem of the bacterial system is misfolded proteins. The proteins are going to get formed into inclusion bodies, So therefore it is going to be required to express the chaperonins, and also people are trying to solve this problem by using refolding buffers. Limitations for expression of eukaryotic protein in case of bacterial system is mainly due to the difference in the codons which appears between the two organisms, which is going to be playing a major role in means expression of the polypeptide chain in the prokaryotic system for a protein of eukaryotic system. Another very important difference is post-translational modifications, mainly glycosylation and disulfide bonds, so which causes accumulation of these antigens like that. Then factors which affects the expression of clone genes means if you go to any of the expression system, expression vector, so these are the factors which mainly involves in regulating the expression of proteins. The first one is promoter strength, then transcriptional termination, plasmid copy number, plasmid stability, host cell physiology, translational initiation sequences, then codon choice and mRNA structures. These are the few components which one need to take care whenever the people want to go for expressing a particular protein in a prokaryotic or eukaryotic system. So whichever we want to go for, we need to have to address these particular aspects, so that all these aspects are to be considered and made suitable for expressing that particular gene in a particular specific host. So this is how it's going to be done. We are going to have a recombinant DNA construct, so which is going to get transferred into the particular organism. Here we are showing equally, you can see there is a plasmid expression vector carrying T7 layered promoter with an G SCF cDNA, and so by using a lac without promoter there will be production of this selection is going to be done, and then the T7 are going to be get produced. These T7 polymerases are going to be used to produce a specific GCSF protein, which are going to get accumulated within the bacterial cell. So it is also possible, people are also doing it, that they are making the bacterial cell or the cell lines to secrete the protein outside the bacterial system or the cell system, so that it will be easy for separation and purification of that particular target protein from the spent media. So this is the steps. First, the gene of interest need to be identified, then full length cDNA has to be get synthesized, then insert it in an expression vector next to a suitable promoter, then clone into a suitable expression system, either bacteria or yeast or mammalian cell lines or insect cell lines, then screen for positive clones which are producing our protein of interest, then select the best construct or the expression host which is producing protein in a little good quantity, then go for, in case of mammalian cell, expand the transient and stable transformants, because here in case of mammalian cells there is a chance of transient and stable transformants. We need stable transformants which can produce this particular protein for several passage numbers or several cycles of cultures. Then further we can go for cell based, that is in mammalian cells. In case of bacteria or yeast or insect systems, directly we go for scaling up, and then we isolate and purify the protein of interest. So in case of bacteria, how things are going to go for, here it is shown. Take the construct, go for selection screening, then transform the cells, get the particular cell, then made it to produce a protein. That particular protein you can go for determine the solubility, then go for study the protein, and then scale up into batch culture, into lab culture, then there is batch culture, then scale it up and isolate the protein of interest from the spent media by the chromatographic method, especially affinity is used, then you can go for further purification by using size exclusion and affinity, and then confirm presence of protein and quantify it. So there's a few expression systems and advantages, disadvantages in the form of table, which you can use it to write in detail. So baculoviral expression system that is being used in case of insect and mammalian cell lines. So first people reduces the recombinant baculoviral vector, then they co-transform the recombinant vector with the disarmed viral vector, because here because these particular viral vectors, they are going to carry all the genes for infection, whereas the recombinant DNA vector will not carry the genes for infection. So they allow them to mix up together and recombine within the particular host cell, where it leads to the production of recombinant baculoviral vector carrying the target DNA molecule, and then further we can use it to infect large cell lines and produce a number of stable transformants for the production of specific protein. And these are some of the commercially and viable proteins which people are producing by using this particular method, and here and which are being used in therapeutic agents in different disease conditions. Here I'll be mainly concentrating on the production of insulin protein, which is done through this recombinant DNA technology nowadays. So insulin, we all know that it's a 51 amino acid having protein carrying two polypeptides produced by the beta cells of islets of Langerhans. It has A chain, 21 amino acids, B chain with 30 amino acids. Both are held by number of inter and intra disulfide bonds. Then how to produce this insulin? So in order to produce insulin, what they are doing is, it's not possible to produce it in the similar fashion like the normal cell produces, because in normal cell, it is in beta cells, it produced as pre-proinsulin, then proinsulin, then to insulin, and then during that conversion there will be cleavage of single polypeptide chain into two, which becomes A and B chain. So here in the recombinant DNA technology, what we are doing is, so people are actually converting this by using the protein that's A chain and B chain. By the using the redundancy of genetic code, they are developing the synthetic gene which can code for A chain and B chain separately, and this A chain and B chain is being kept into a laxed gene under a specific promoter in a particular expression vector having ampicillin resistance gene as an target. Then they are transforming into the E. coli, and these E. colis are being selected for producing A chain and B chain, and those E. colis which are able to produce, they are being screened, selected, isolated, and then further these particular cells are going to made to produce the polypeptide chains of in large quantity, and then they are separating these particular A chain and B chain separately. Then they are subjecting it for because these particular proteins are going to get produced, these polypeptides are going to get produced as a, what I can say, a fusion protein with the laxed protein. So they are going to be made, they are going to place this particular gene such a way that they can be cleaving out this specifically this particular A chain and B chain. So first they are going to isolate and purify the fusion protein. Then isolated fusion protein will be subjected for cleavage by cyanogen bromide. This CNBR can specifically, as we know, can cleave at the aromatic amino acid, so it can be used to cleave that, and then these A chain and B chain, which separately will be get obtained, will then further they are going to get mixed together and then oxidized with the cysteine, and then they'll made to get produce the disulfide linkages between them and intra and inter, and then they obtain the mature insulin molecule. So further this particular insulin is going to be evaluated for its activity, and then it is going to be used for therapeutic, as a therapeutic agent. So the next type of protein which I thought like it will be helpful is vaccine, recombinant vaccine protein. So here in case of hepatitis B, people are producing the recombinant vaccine for hepatitis B, which is being produced by using yeast as an expression system with the ADH1 promoter. So this is the one. They take this hepatitis B A antigen and then cut it with the required one, and then they are glycating within the yeast expression vector is plasmid MA56. Then that particular and it is being placed at alcohol, and there is ADH alcohol dehydrogenase promoter, and then they are going to get transferred the yeast system, transform into the yeast cells, select the yeast cells based on the tryptophan as one of the marker they are using as an absorptive strain. They use it and they select it, and then those particular cells which are able to grow with the yeast tryptophan, they are going to be isolating, and then it will be sedimented. They are going to use it for the production of the hepatitis B A antigen genes. So here those particular cells which are going to be producing this particular hepatitis A antigens are selected, they will be lysed, and then the antigen protein is going to be purified and it is used commercially. This particular method of recombinant DNA technology is also being used in case of plant systems. They are using this particular method to produce varieties of transgenic plants, that is like insect resistance, stress resistance, or they are producing the plants which can produce with an augmented quality like golden rice, or insect resistance like BT, cotton. Okay, so like that people are producing varieties of transgenic plants. So first of all, these transgenic plants are nothing but those plants which carry the gene of foreign origin of other organisms. They are called as transgenic plants. So Monsanto and the CSIRO of Australia together, they have produced this killer cotton, which carries this particular gene from the cry gene of Bacillus thuringiensis, which was identified to kill this bullworm, which is a common pest for the particular cotton plants. This toxin is found not to does not harm the spiders, humans, and other mammals. So the Bacillus thuringiensis endotoxin gene, that's cry gene which is used here. So how they did it, they took the cry gene and they inserted into a bacterial vector. Then that vector was that's a plasmid, Bacillus plasmid. Then the vector was made to get transformed into an agrobacterium species, where they made this particular vector to get recombinated with the R plasmid, RI plasmid of agrobacterium rhizogenes, and then they transformed this into an TI plasmid of rhizogenes, tumefaciens, and then they put it into a bacteria. This agrobacterium tumefaciens was selected, which was carrying the recombinant vector. Then they infected it into an particular desired plant protoplast. So those particular protoplasts which got infected were selected, and then those particular plant means those were developed into a transgenic plant. And here they introduced a control plant and allow the larva to infect it, and they found that it was resistant to that particular pest. So this is a method people used to produce a killer cotton plant. Then into transgenic animals. These transgenic animals are those animals in which, again, the foreign gene or foreign gene of foreign origin or the other organism is being introduced. Uh, People have used this transgenic technology to produce transgenic mice, where people are using method like retroviral vector DNA, microinjection, genetically engineered embryonic stem cells, Kralox-P recombination system, or high capacity vectors, and they are also producing this transgenic mice for Alzheimer's, and as test system, conditional regulations, and to control the cell death like that. People are also producing transgenic cattle, sheep, goat, pigs, birds, fishes, varieties of organisms with the transgenic technology. So how it is done in case of transgenic mice, I'm showing here, which is being used in other cases also. They are producing a retroviral vector construct carrying that carrying the transgene or the target gene. First, they take a donor female. They isolate the embryo of 8X cell stage. Then they infect that particular embryo with the retrovirus. Then that infected what you call it as embryo is going to get implanted into a female. So now this female is going to produce the progenies, out of which few of them are going to be transgenic, which we can identify based on the marker gene. So people are also doing another method, that is microinjection method, where they are isolating the fertilized egg, and then to the fertilized egg they are going to micro inject the transgene, and then that fertilized egg is going to be implanted back into the female, and then they are going to check for the efficiency. So here expression will vary in transgenic offspring due to position effect and copy number. That means where exactly the gene of interest goes and get itself inserted into the main genetic material or into the chromosome, and as well how many copies of it are going to be there. This is another method wherein people are taking the egg and the sperm from different organisms together, they fertilized outside, and then they go for micro-injecting technology, and then they produce the transgenic mice. The another very important method which most commonly being used is also genetically engineered embryonic stem cells. So here what they do is they are going to isolate an embryo from a donor organism. The donor embryo is going to the blastocyst is taken, the embryonic cells are separated. These stem cells then they are going to be transfected with the transgene through a vector viral vector system. Then these transfected embryonic stem cells are going to be selected, enriched, the transfected embryonic stem cells. Then they will be further again put back into the blastocyst, and that blastocyst is going to be again implanted into the female, and then which is going to give rise to the transgenic mice. So the method are being used to produce varieties of transgenic mice, knockout mice, and many varieties of diseased or susceptible or resistant like that, which are being used in varieties of scientific studies. Then we come across with transgenic cattle, wherein they are making this casein gene to produce to become means they are putting extra copies of casein gene to produce 13% of more milk protein into the milk. By using the same methodology, they have produced enviro pig. So the pig which is producing phytase in their salivary gland, which can eat upon the phytic acid, can be used by these particular pigs. Then they are producing transgenic fish, the tilapia, salmon, catfish, many have been developed. So with the extra copy of growth hormone, they have made these particular fishes to grow more and accumulate more amount of flesh in them. People are also using this transgenic technology to produce animal bioreactors, or we can call it as farming. So wherein they are making the sheep, for example, here to produce more of protein, and they are also making the sheep to produce a specific protein into their milk, like alpha 1 antitrypsin, treatment of emphysema and cystic fibrosis. They created a sheep which is called Tracy, producing it in her milk. And they have also created this silk gene. They have transformed means into the from the ops spider into the goat, and resulting in the male goat were used to Sere silk, producing female goats. Each goat produces several grams of silk protein in the milk, and then extracted silk protein can be used as dry powder and can be spun into fiber. GTC Biotherapeutics has received approval for producing to sell the antithrombin protein produced from the goat milk, like that. So this is called biofarming or farming, which people are also being used, which is an extensive application of recombinant DNA technology. Thank you. In case further in doubts if you have, please do contact. So with this I can say the syllabus for your batch from my end is done. Further any help you need, you can contact me. Thank you. All the best.